When we study cell as the basic and fundamental unit of life, we come across the term biomolecules, which compose its cellular pool and are important for the functioning of cell so that it acts as the basic unit of life. So in this topic, that is of biomolecules, we are going to come across few terms like what is cellular pool, what comprises the cellular pool, which are the molecules basically. Now I am going to tell you what is the cellular pool. They are the molecules which are present inside the cell, which are responsible for carrying out the various metabolic functions. So we have in cellular pool few inorganic materials and few organic compounds. Now what I need to tell you over here is that cell, which acts as the basic unit of life, that is quite repetitive. We know that the basic functions which distinguish a living organism from a non-living is the term metabolism. Now what is metabolism? Metabolism is sum total of catabolism and anabolism. Now what are these terms? These terms are nothing if they are not in reference with the materials which are inorganic materials and organic compounds. Now how do we distinguish that something is living and something else is non-living? Because a living component will perform certain functions of breaking certain compounds and making few compounds out of those compounds, so that there is a continuity of life that is being maintained. The various functions which ensure that the cell is living is carried out in the process of metabolism. Now what are these compounds which are taking part in metabolism? They comprise the cellular pool. So basically we see that biology has in its root somewhere chemistry which is being involved. The chemical constituents are involved, their size, their composition is made in such a way so that they carry out the living functions, various living functions which distinguish a non-living chemical substance from a living chemical substance. Are you getting my point? What I am trying to explain is that in the living substances or in the living cells, we have molecules which interact with each other in such a manner so that living process of metabolism is carried out effectively. So we are going to deal over here with the cellular pool components, which are the inorganic materials. So let us see which are the inorganic materials first of all. The inorganic materials include many salts which are present inside the cell. It includes mineral ions and of course the universal solvent, that is water, having a high latent heat of vaporization and other properties which makes it a suitable solvent for the purposes of metabolism. Water is very important. Then comes the mineral ions. We have studied earlier that mineral ions are very important in various metabolic functions, like calcium ions are important for opening up of the pores of the membrane, and salts, of course, they also play an important role, because the mineral ions which were important for the plants, you might have read or studied somewhere, those mineral ions have important role in any cell. In fact, these are micro elements and macro elements. We have mineral ions in macronutrients under the category of macronutrients and micronutrients. They are taken up. They are inorganic material which the cell needs to take, and they might form ergastric substances of the cell. What about the organic compounds? There are four major classes of organic compounds that we come across in a cell. First one being carbohydrates. If ever you haven't heard about this term, these are the sugars that we consume in our food. Then the second one is proteins. Third one is lipids. And fourth one is nucleic acid. So these four organic compounds, they interact with each other. They have such characteristic properties which enable them to carry out the functions of catabolism and anabolism and maintaining the cell as a living entity. These four organic compounds are responsible, along with the salts, mineral ions and water, which are transported through and across the cell so that the cell maintains that living status in itself. Now the question is that how the macromolecules are formed and what are macromolecules. For that first case, that what are macromolecules? Macromolecules are those molecules which are formed of the organic substances when they form polymers. They are large size molecules which undergo some change in their shape. The shape is formed in such a manner so that whatever reaction is required, I am telling you in context of these four, that these macromolecules, these are large size molecules, organic molecules, they have such arrangement and composition so that they form certain crevices, their specific shape and structure is formed, and in that process they carry out the activities. So the macromolecules are often formed by polymerization process where many molecules come together and they undertake the activities whichever are required from them. Then we are going to discuss carbohydrates in detail in this lesson. So let's take into account that what are carbohydrates. When I use the term carbohydrates, I am referring to the macromolecules which are made up of sugars. Okay. Now when I use the term sugars, then there is another term which is used, that is saccharides. You might have heard these terms in your chemistry study. Saccharides are there. Now carbohydrates are of three types. There is a classification, broad classification of carbohydrates. First one, the first type of carbohydrate is, because I am talking about saccharide, so it is monosaccharide. Okay. Then the second one is disaccharide. There is another classification known as oligosaccharide, but we need not to get into details of that. And the third one is polysaccharide. Now, for understanding what are these three, we need to know that what is the basic structure of a sugar. If I am talking about a saccharide, what would be the basic structure of a monosaccharide? I am considering glucose over here. Okay, glucose is a monosaccharide that you hear about, that it is the basic sugar which is present, which is formed as a result of photosynthesis, and then it is converted to other forms, and then it is circulated in the entire food chain. So what are carbohydrates? Carbohydrates are nothing but the term that you have to remember is polyhydroxy aldehyde or ketone. So what you have to remember, that there would be a long carbon chain, or there would be a cyclic carbon chain with many hydroxy groups. Hydroxyl groups would be many, and there could be presence of either aldehyde or a ketone that would make a sugar, as in the case of glucose. So if we have to understand what is the empirical formula of carbohydrates, you know that it is CH2O, and this happens to be the empirical formula of any carbohydrate that we take into consideration. And if it is to be understood from the stereochemical aspects, it is polyhydroxyaldehyde, that there would be many hydroxy groups and there could be either aldehyde or ketone group, and this polyhydroxy group could be present either on a straight chain or it could be present in a cyclic carbon ring. So the empirical formula happens to be this, and you know that glucose is C6H12O6, which is well suited with the empirical formula. Now in a monosaccharide you find only single polyhydroxyaldehyde ketone, and when we talk about disaccharide, we have examples like maltose, we have sucrose and lactose. Now these are disaccharides. Now as the name suggests, it has di means two. The two sugars which are present in case of maltose is, maltose has glucose and glucose. It is a disaccharide of two glucose molecules. Talking about sucrose, it is galactose plus glucose, it is glucose plus fructose. Okay, and talking about lactose, which is milk sugar, that is galactose and glucose. Alright, so you see over here that two monosaccharides are getting connected by a glycosidic bond. Okay, Now there is a glycosidic linkage present between the two sugars. This term is to be remembered. First thing that was to be remembered, that is polyhydroxyaldehyde or ketone would form a carbohydrate. Three types of carbohydrates are present. First one is monosaccharide, disaccharide, and there is another term oligosaccharide which includes 9 to 10 sugars, and then there is higher term, that is polysaccharide. So when we are studying disaccharides, we come across a term known as glycosidic linkage. Now this glycosidic linkage is present between the two monosaccharides which form a disaccharide, and such would be the case in the case of polysaccharide as well. So the glycosidic linkage is basically the dehydration synthesis that takes place between the molecules. Okay. This dehydration synthesis leads to the loss of a water molecule because carbon of one of the sugar would get bound to the oxygen of hydroxyl of this sugar. I am repeating it, the carbon of one sugar would bind, this supposedly this sugar gives its carbon for binding with the hydroxyl oxygen. Okay, so this bond would be formed, and in return there would be a loss of water molecule which leads to dehydration synthesis, and a glycosidic linkage is formed between the two sugar molecules. So this is all about disaccharides. When we talk about polysaccharides, we have again a lot of variety of polysaccharides which are present in biological world, and when we come across these examples, we get to understand, or we get an idea that what would be the function of the carbohydrates in a cell. So let's take the example of polysaccharides first. Polysaccharides would have huge chains, like supposedly we talk about cellulose. Cellulose is very much abundant in the plant world, in entire world. This cellulose has almost 1000 or 10,000 molecules of D-glucose. Okay, so it is a polymer. Okay, when we talk about polysaccharides, we talk about homopolymers or heteropolymers. You see, we have large polymers, many monomers are involved, and what are the monomers? They are sugar molecules like glucose, and we have heteropolymers. When I talk about homopolymers, I gave you the example of cellulose, and when I talk about heteropolymer, we are going to take into account agar. You have read about this agar. This is taken from algae, sea algae. This is used as a lab material in many places for biotechnological processes. So this is a heteropolymer basically, alright. Then we have cellulose, which is homopolymer. Then we have peptidoglycans and many types of polysaccharides are present which are important for the building material of the cell. Okay. Secondly, they are storehouse of energy. The linkages which are formed, they have a lot of energy stored in them. So we have carbohydrates or sugars as the energy source for the cell, and we get to understand the three types of carbohydrates which would be present, the function that they would be performing. First one is the energy source, second would be the building material. So this is all about carbohydrates. We are going to take into account next lipids.